biology are you interested in, and how would you describe it?
I've always been interested in cancer biology. I did my postdoc in a cancer biology lab, but mostly because cancer biology is really just normal cells that have gone wrong. So we learn a lot about what a cell should do by learning what goes wrong when cancer's developing.
Do you do research in your role in the university?
So my primary role in the university is teaching. We're very much a teaching-centric university. However, there is an increasing need to do what I would call mentorship research. So research that isn't going to be published necessarily, but students right now I've found have such a hunger for research. They want the basic process. So I've taken on usually about 5-6 students a semester that will have interests in a couple of research projects that I do. One is a cancer biology project, and then one is actually more molecular based and a little bit easier to accomplish in the time frame that we have available between classes. But yeah, these students, they just want to do the basic research. They want the mentorship with me, the direct relationships. So I do every semester do a bit of research, yes.
Yeah, I'm interested in researching when I go into biology or marine biology.
Yeah.
So I'd very much be interested in like a research-based role. So
yeah,
it's interesting to hear your perspective on that.
Including students on it, including them in collaborations with other departments is something that is a really good, sets them up well for academia because collaborations are how we get by. So we collaborate with psychology and other departments, and it really is effective at getting students the process of science research. This is our cell biology lab. So what we're looking at here is cancer cells from a human patient with breast cancer. This is something my research students are doing. So what they're going to look at is when you grow cancer cells in culture, they like to grow with serum, like all the nutrients and things like that. A lot of cancer cells can grow without serum. So we're reducing the amount of serum and seeing if the cancer cells can still survive, which normal cells would not, but these cells seem to be able to survive even without it. They're very resilient, which is the problem. That's legitimately when you were asking me earlier what students are doing for research. This is the kind of thing that we'll do. So we'll starve them of serum and see how long they can survive.
What inspired you to teach?
That's a great question. I did not go into my graduate program thinking I was going to teach. I was very interested in biology. That was it, period. So I went through the graduate program. I did what everyone sort of does after that, which is I went into a postdoc in San Diego in the Cancer Research Center at UCSD. So I was doing a cancer research project on how cancer cells metastasize to secondary locations. And I had to pick up some samples at the cancer center just to do some microscopy on. And I went to pick up those samples and I saw these patients being wheeled in by their significant others or family members. And I felt like the little thing that I was doing of basic research, it just felt so far from the clinic, and I felt like I was making zero difference at the time. So I realized at that moment that what I really need is to be able to impact people a little bit better. And all I had done was academic research, basic research, cancer research, and never explored outside of that. So I decided on a whim to leave my postdoc. I was planning on coming back, but I just was going to take a summer away and teach in a part-time role at a community college, and I absolutely loved it and never looked back. I was like, this is my calling, this is what I meant to do. So it wasn't my original goal, but it really sort of, I was led to that. So what we would do is we would take a sample from a human patient, and that would be the DNA template. We would put that in here into these, not really liquid, but put it in here. So, yeah. And then add the enzymes and the buffers and everything needed to basically do DNA replication in a test tube. And then when we're done with that, all of the ingredients are present in the tube. Put this in, and these put this in the PCR, and let it run on its own, and it'll go to all the temperatures it's needed to go to get the DNA replication to happen. And it does it over and over and over again until you start out with 2 DNA molecules and you end up with something like 8 million.
Wow.
So you have enough to actually visualize and see whether that DNA was present in the solution or not.
Wow.
Yeah.
That's super cool.
It is, actually. It's amazing technology. The Nobel Prize was awarded for it.
Wow.
Because it's really advanced science, diagnostics, forensic science. We all use this technique. So PCR
Wow.
is really common.
What's your favorite life form?
Life form. Okay, well this is gonna sound bad, but I actually have a very big respect for viruses because, again, speaking about using a basic cellular process, in their case to their advantage, to evade our immune system, and they're not actually technically even life forms. They're not even living, and yet they have found, I'll put that in quotes, mechanisms to get around our immune system, which is tremendously complex. They figured out ways to continue to reproduce, to continue to survive, to continue to beat out our immune system, constant competition with our immune system. So I don't know if I'd say favorite, but I have a very big reverence and respect for viruses because of that.
What's your personal relationship with the creatures you have studied?
Personal relationship. I think for a while it was very easy to not have one. Coming from a background of molecular biology, what we often do on a daily basis is, and this is something that I've had students say to me as well, pipette something clear into something else clear. And it's really easy to just get lost in the minutia of that and not think about what you're actually doing. So for a while, I think I didn't think about that. It really wasn't until my experience with being a postdoc and going to the cancer center and seeing, wait a second, what I'm pipetting of clear solution into clear solution is actually something that is driving this cancer. So I think it just took me a while of building my career before I actually got any relationship to what I was doing, and then it's hard to turn off a little bit. So now as I teach, I do a lot of disease-based models. And again, I still teach molecular biology, teach cell biology, so we still can't see what we're talking about, but I very intentionally add these diseases as saying, yes, maybe this is a minute process that you can't even see, but understanding that allows us to treat this illness in a better way than we ever thought we could by understanding it. So I think it's developed throughout my career rather than started out that way.
Yeah.
It looks like these are GFP, which stands for green fluorescent protein, expressing bacteria cells. So they are cells that have been given the GFP protein, meaning that when they're exposed to UV light, they'll glow. And that's what we're seeing basically here. You can see that they're kind of green compared to the rest of the plate.
Are there any questions I haven't asked you that you think I should ask you if you were in my position?
I think a question that I'd love to answer to a student thinking about pursuing biology would be, what are some alternative paths that aren't always presented to you as a student as going through your training? Because I feel like those weren't necessarily presented to me. And so to that I would say, it doesn't have to be the conventional going from grad school to postdoc to industry or academia. It could be so many other things in between there. There's a lot of practical science, forensic science, these areas that sometimes aren't presented as options, science writing, even science illustration. I have grad student buddies that do that was never presented as an option. And so if I were starting out as a biologist in my career, I would love to know that these are all in front of me, and maybe I don't know which one I'm going to follow, and maybe that'll emerge throughout my career, but I wish that I had known these were options.
Yeah, even like filming biology.
Totally, I didn't use that example, but yes, absolutely. Yes.
Thank you so much
You're
for your
welcome.
time.